Right, I'm going to switch our attendees and allow them to
Yes.
promote to a panelist. So would you like to do the introduction?
Yes. Actually, I don't have to work very hard because this is the first time ever in which a speaker actually gives me a small paragraph of how to introduce the person in principle. So it's interesting because obviously a lot of people in IMO are going to know Artem. Artem needs little introduction. He's actually someone who worked at IMO after we, and when I say we, I mean Jacob, Sandy, and myself managed to kidnap him from McGill University, where he was doing a master's, and brought him to Tampa. He's hoping that as a Canadian, he liked the place, and I think he liked, especially the department, or at least mostly the department, and the conversation with other mathematical oncologists. He's not a mathematical model, though. He describes himself as a computer scientist and someone who is trying to look at biology through an algorithmic lens. He is clearly someone who is very interested in our reads. His blog is in the bottom of this presentation, often updated with really sort of well thought thoughts on mathematical modeling, often about cancer, not always. A few years ago, he decided to do a DPhil in Oxford and left Tampa, kept the affiliation with Jacob when Jacob left for the Cleveland Clinic. So he's got dual affiliation right now. And I'm really intrigued to see what Artem is going to be talking about today. So Artem, please take it away.
Great. Thanks for inviting me. Happy to be always back at Moffitt, even if it's virtually. I can only see Sandy, so I'm going to use him. Oh, I can't even see Sandy anymore. Hopefully everybody can see the slides. And if I draw on them, can everybody see a smiley face I drew in the corner? Yeah, okay, good.
Yes, I can see.
Great. So I'm going to talk about how to measure the eco-evolutionary dynamics of cancer and other microscopic systems. But really, I'm just going to talk about one sentence. So there's this sentence. There are only 26 letters in the English alphabet, and yet there are more than 26 letters in this sentence. It's taken me probably 12 years to understand this sentence, and I probably still don't fully understand it. But I think by thinking about this sentence, we can become much better cancer modelers. And in particular, I think we can do much better work in using evolutionary game theory to talk about cancer if we really spend some time to think about this sentence. So just keep the sentence in the back of your head as we go through the talk. I'm not going to take you through the whole 12 years of making sense of the sentence. I'll just talk about 3 papers that try to, in some grand arc, make sense of this sentence. I'll start with sort of what I think is a typical use of evolutionary game theory in oncology, which will show us sort of one reading of this sentence. Then I'll ask, but how do we know the game in that particular typical use? And suggest that maybe in the future, we shouldn't rely so much on intuitions about these games, but we should actually just directly measure these games. So let me start with a typical use of EGT in oncology. And here I'm just gonna, there's many papers you could pick. There's a lot of great papers. I'm gonna pick on one of my own just because I know it well. And if I say mean things about it, I'm just saying mean things about myself. So in economic game theory, there's sort of 4 main components. There's utility, there's the player, the strategy, and all of that is summarized as the game. And in economic game theory, we think of utility as something that's given to a player based on that player's strategy, which resulted from some rational choice process. Mm-hmm. And that whole thing is summarized as a game. Right? So how do we make sense of, how do we usually adapt this in a typical case to the evolutionary setting? In evolutionary setting, we replace utility by fitness. And we say utility is a property of a token fitness is a property of a token organism based on its phenotype, which in game theory terms could be thought of as a pure strategy, which is in the simplest model is fixed from birth. And all of that is summarized as, again, people usually say a game or an evolutionary game. I'm going to start calling it a reductive game and we'll see why more later. But this is, I think, the typical interpretation of games in the evolutionary setting. So let's look at a typical use of evolutionary game theory in oncology. This is an old paper of David's, back around the same times as I started thinking about that sentence. David considered, two cells. Some of these cells are invasive cells. Some of these cells are AG or autonomous growth cells. They inhabit, some sort of, tumor environment. If two invasive cells, so invasive cells try to move always to find new resources if they don't have them available to them. Autonomous growth cells just try to use all the resources available to them to grow as quickly as possible. And so if two invasive cells interact, each one has a 50-50 chance of moving. And if it moves, it pays some cost to move and then gets some benefit of finding a new resource at the spot, and the other cell stays at the spot and gets the resources. So the expected fitness gain for them is 1/2 B plus 1/2 B minus C. On the other hand, if invasive cell means an autonomous growth cell, then in that case, the autonomous growth cell never wants to move, so it gets the whole benefit B of just sitting there and eating all the resources, while the autonomous growth cell has to move away before finding resources, so it pays, the invasive cell has to move away before finding resources, so it pays the cost C before getting that benefit, getting a total of benefit of B minus C. And then if two autonomous growth cells interact, since neither one can move, they have to share all the resources at the spot, and so they each get about 1/2 B. So this is, David called this the go versus grow game, and has gotten relatively popular. So what would a typical EGT study of this look like? Well, let's look at our study of this. We said, okay, in the original treatment, David just imagined that all of these cells swam around in a soup and interacted randomly, but that's not what happens in a tissue. In a tissue, they're involved in some sort of spatial structure, and when a cell dies, the only cells that can actually repopulate the newly vacated spots are the cells next to it. And thus, those cells themselves only get their fitness from also local interaction. So for example, this cell here that I just drew a box around only really interacts with the brown cells, and maybe this blue cell as well. And so its fitness comes from here, while this blue cell down here, which I'll put a triangle around, interacts with these pink cells, and maybe this blue cell as well. And so its fitness comes from these interactions. So this gets a notion of locality. And we can take this minimum notion of locality and see that this corresponds to a particular transform called the Otsuki-Novak transform, which lets us take the go versus grow game down here, plug it into the transform, and see what space does to it to get a new game at the level of the population, at the level of how the two types of cells are represented in the whole population. And it in general makes the game more complicated, but we can still get the transformation. So the idea is we start with some intuition about the lower level. We combine with some other intuition we have about, let's say, space. In this particular case, we started with two very simple intuitions, so we could combine them formally and mathematically to get some result at the population level. And I think this is the usual direction that we'll see in sort of a typical EGT in oncology study. And
Before
afterwards,
you
we might.
Artem, can I just briefly ask about that? Because that transformation that you made from a non-spatial game to some sort of spatial interpretation of that, what exactly is the spatial structure that transformation assumes?
So I don't want to focus too much on the details of this particular case study. In general, you can imagine your favorite transformation. So you can imagine your favorite, off-grid hybrid model with diffusing things, etc. And that will also be some transformation from a local description to some global. Just in the more complicated case, we won't have a simple mathematical formula for the transformation. It'll be implicit transformation, and we'll usually get it through simulation. In this particular case, we get, just a simple transformation. So the details of the transformation aren't essential. For the particular case of the Otsuki-Novak transform, if you're interested in that, if you want to interpret it in terms of graphs, which is how Otsuki and Novak wanted to interpret it, then it's exact for infinite Bethe lattices, and a pretty good approximation for k-regular graphs. And the reason it's pretty good approximation for k-regular graphs is because locally k-regular graphs look like infinite trees. So that's why it works there. But that's not the only way we can interpret this transformation. In this particular paper, if you go through it, you can see that we talk about this transformation as a general first order approximation of space. So you can imagine taking space and decomposing it into interactions, into correlations that build up between cells and their neighbors and their neighbors' neighbors and their neighbors' neighbors' neighbors, and so forth and so forth. And if you truncate that approximation at the first step, so the correlation between cells and their neighbors, then you get this transformation.
Okay, so I understand you don't want to sort of focus too much on this, but I guess my kind of retort to that is, why not just build a spatial game if you're going to look at the impact of the space, right? And the idea here is that maybe this approximation does kind of do that, but is it effectively adding like one extra nearest neighbor interaction? Is that kind of what you're implying? Or is it more general than that?
Yeah, it's adding the structure of in this particular case, the reason we did it in this particular case is because we just wanted to look at the sides of the interaction neighborhood and how that affects, and this is sufficient to get you an interesting effect from that. Okay. But that being said, again, if David has done versions of this with, more complicated spatial structure and, lattices, etc. The point is that in that case, you would have still some spatial structure that you're plugging. This would be the interaction payoffs between two agents next to each other in whatever spatial structure you gave. And then you still run it through the spatial structure, and that would still produce some other thing happening at the level of the population. It's just in the general case, we wouldn't know what the effective game looks like in terms of the reductive game, because in the general case, people don't know this as this transformation as an equation. They just know it implicitly as a simulation. But again, this is not going to be central to the thing. I just want to give this particular example because A, I know it, and B, the formula is simple, so we can actually see how the game changes explicitly. But yeah.
Okay, thanks.
So yeah, when we do structure, we usually we'll link this spatial structure to something. And as Sandy was asking, in this case, what do we want the spatial structure to capture? Well, we wanted it to capture the difference between neighborhood sizes in the bulk of the tumor, where you're interacting sort of with lots of neighbors around you, versus when you're up against some static boundary, like let's say against a fibrous capsule or up against a blood vessel, where your neighborhood interaction neighborhood is inherently smaller because half of your space is kind of cut off by that hard edge. And you can see these sort of hard edges if you look at here, some imaging data here. This local space structure looks different near boundaries versus in the bulk of a tumor. And what do? We then said, okay, different kinds of neighborhood sizes correspond to different number of neighbors in this transformation. And something like the k=5 line here would correspond to being up against a tumor boundary, while this k=8 line would correspond to something like being in the bulk of the tumor. And if we then make the phase space of possible dynamics, we see that it could be that you have one set of dynamics at the boundary and another kind of dynamics happening in the bulk of the tumor, which of course you can then link to worries about measurement in actual cancers by saying, well, if you're doing something like a biopsy, you're getting mostly samples from the bulk of the tumor because the edges always, edges are always smaller than the bulk. So you're always getting more of the bulk. And so it might be hard, you might be in this mixed equilibrium state here at the edge where it matters most, where you don't want invasive cells. And yet when you sample, you'll get mostly bulk. And so you'll mostly get autonomous growth and you'll think the tumor is benign when it really isn't. For example, again, the particular conclusions you draw from this are different depending on the typical study in EGT. This is just in this study. And then usually people then go on to compare these observations to, you know, to some data, usually at a very qualitative level. So if you look at an image like this staining on the left, you'll notice, and I'm not going to go into how to read the stainings exactly, but you'll notice that near boundaries, you end up having much more mixed genotypes as we would expect, while in the bulk, you end up having much more monotypic populations. But of course, how is this useful? How do we know this game? You know, when David first defined the Go versus Grow game, how did he know that that was the game to define? And in particular, let's just look at the pipeline of your typical EGT study. How does it go? It goes with some intuition about a game, in this case, Go versus Grow. It then looks at some important aspect that we care about, in this case, the difference between bulk versus edge of tumors. It then conceives another intuition of how that might transform the game. It then transforms the game and compares the outcomes of that game to some, you know, usually qualitatively to some data. And that's kind of, you know, a typical pipeline. And what I'm suggesting is that we should try to invert this pipeline. Instead of starting from this bottom up, we should start with careful measurements of the effective game. And then if we do want more reductive games, think about how do we carefully measure and represent spatial structure in order to project backwards towards the reductive game. So, and this is where having nice transformations really does matter. So why did I pick this transformation? This transformation is easily invertible. In the more general case of that Sandy was alluding to of some complicated spatial structure that we usually study through simulation, will be in a case where even if we know what the effective game is, we don't know how to sort of transform it backwards necessarily through the spatial structure towards the cell-cell interaction level. So let's come back to our to our different interpretations of economic game theory. And in particular, so as I was talking about the typical study, we talked about fitness. It was a property of those individual cells that were either staying put or moving around slightly. It was based on their strategy of either being invasive or autonomous growth. Their strategy was fixed from birth, etc. And let's revisit the sentence that we started with. So there are only 26 letters in the English alphabet, and yet there are more than 26 letters in this sentence. So does anybody want to volunteer how to make sense of this sentence? Why is it not a contradiction?
Spatial interaction generates more heterogeneity than like singular or non-spatial individuals.
How does that relate to English letters?
Well, that the letters on their own have only 26 different sort of morphologies, but when you put them together, you get a lot more stuff going on.
Okay. Yeah, I think that's an interpretation. I think the interpretation that Pierce was going for is similar to that. He would point to the word letters being used in two different senses in this sentence. When we say there are only 26 letters in the English alphabet, what we're actually saying is there are only 26 letter types in the English alphabet. And when we say, and yet there are more than 26 letters in this sentence, what we're saying is there are more than 26 letter tokens in this sentence. And this is a very useful distinction between a type and a token that I think can make us a little bit better at how we make sense of evolutionary game theory models.
Hey, Artem.
Yeah.
You had more time to think about the words you're choosing here, but just to bring us up to speed, how would you define in this context type and token?
Oh, I don't think there are good definitions of type and token. So type and tokens are examples of types. and types are things that there are many tokens of. So in the case of this particular sentence in front of us, the types are, the E's are a type, and we can see a token of the type appearing here. We can see a token of the type appearing here, a token of the type appearing here, a token of the type appearing here, and so forth.
So
that
right, you're using the same words to explain the same word. That's not very helpful. So
yes.
So my question is, it really is, so I just don't think that type and token, the way I understand type and token, would really improve the meaning and understanding of that sentence. So that's why I mean, you can write exactly the same sentence that there's only 4 base pairs, yet your DNA is much, much longer, right? So there's a context, right? You have basic construction pieces, like letters in an alphabet, but in context, they combine to make something much bigger. Or you have black and white keys on a piano, but they sound remarkably more colorful than just 2 colors. So I'm not sure if, so anyway, sorry, I stopped talking.
I don't think the point here is to get at the context, because we could also do like the letter E and the letter E, the word that I just wrote of a bunch of E's. There's still a sense in which there's only a single letter in that word that I wrote, but there's another sense in which there's 1, 2, 3, 4, 5, 6, 7, 8 letters in that word that I wrote. And the importance of it isn't context as much as there is 1 letter type and there are 8 letter occurrences or tokens. So that's the distinction I want to get to. And I know I'm not giving you a definition of type and token, because in some word they're sort of atomic objects. I can't give you good definitions of them. I can just give you more and more examples. So for example, you could say something like, I am a particular organism, so I'm a token. But at the other hand, I have the type of being green shirt wearing, at least today. So that's the kind of distinction that I'm trying to get at with type versus token.
Fine.
Maybe it'll become more clear as I go into more particular examples. So let's go back to evolutionary game theory and now provide a second interpretation of evolutionary game theory, which loads on the 2 different views of fitness. There's the idea of fitness as a property of tokens. And this is something like where we think of fitness as the number of offspring that a particular individual has. That would be a token notion of fitness. On the other hand, there's type notions of fitness, which are properties, usually statistical summaries of some type of organism. So when we think of something like the doubling time in a basic bacterial growth experiment, that's a property of that cell type, not of any particular individual cell. Now, in certain experiments, there's a clear mapping between the type and token fitness, but as we'll see later, that's not always the case. So what happens if we take type fitness as our definition and run it through our mapping from economic game theory to evolutionary game theory? Well, in that case, we get that fitness is a statistical summary property of a type-structured population of organisms based on the population's distribution of phenotypes, which results from replicator dynamics, and all of that is summarized as the effective game. And so here, the mapping of the words changes a little bit. Now, the player doesn't map to a particular token. Instead, the player maps to a type-structured population. A strategy doesn't map to a particular phenotype, but rather the distribution of phenotypes in that population. And now the population really does have a deliberation process that it's following, which let's say is replicator dynamics. And then so for these cases, I want to talk about that. When we're talking about this kind of interpretation, I'll call that an effective game. And when we're talking about this sort of interpretation, I want to talk about a reductive game. And so if we go back to the mapping here where I used reductive versus effective game. When we were talking about two different cells in the reductive games, we were thinking of two particular cells interacting and what payoff would those cells get. But when we're looking at the population levels, we're looking at how does the proportion of autonomous growth cells or invasive cells changes, and that becomes a type level property of all cells of the type invasive, which are comprised of all the tokens that are invasive. So why is this useful? It's useful because the token interpretation of fitness corresponds much more closely to how a lot of modelers think. In particular, people who are very comfortable with agent-based models usually think in terms of tokens, because the basic atomic objects of study for them, the things that they have sort of direct access to, are the individual organisms or agents, and they usually define what happens to them based on interaction with other individual agents. And so it's natural to define fitness in a token fitness sense. So for example, whenever you have a model where you define fitness of a particular agent as, let's say, the probability to reproduce, or the number of offspring they'll have at the next iteration, and things like that, that's usually a token definition of fitness, and that's the one we're most comfortable with. But in a lot of experimental settings, especially in microscopic systems, what the experimentalist observes is not the fate of a single token, or even the fate of many individual tokens. Rather, it's almost always the fate of a particular type. Because they often can't differentiate individual tokens of that type from each other, or do not track individual tokens of that type. So a classic example might be, I grow 2 cell types on a plate for a while. At the end of my experiment, I take a few of them out and I smear them on a plate and see how many colony forming units come out. That's a type notion of fitness, because it wasn't the fitness of any individual particular cell. Sure, those factored into the type fitness, but that's not what we actually measure. We measure this type level. And why is this useful? It's because we can use this to directly measure effective games. So don't use our intuitions, just measure the effective games. So we can start with, so I want to discuss a particular example of this. This is our the game assay that was in the title of the talk for measuring particular effective games. So in our case, we start with 2 cell types. In this case, 2 kinds of non-small cell lung cancer, ones that have been evolved to be resistant to alectinib, and ones that are sort of never really experienced alectinib. We plate them in a plate, and we might look at them under time-lapse microscopy. So we have some image, and the magenta cells in here are of one type, the cyan ones are of a different type. Maybe we get, for that particular image, we do some computer vision and we count, what exactly are the population sizes of those 2 types, and we'll plot them on a plot, come back at a different time point. Maybe that was at 20 hours, now at 100 hours we come back, do the same thing, repeat that every 4 hours for 5 days. And from those mappings of how the population changes over time, we get an idea of a measure of fitness by doing lines of best fit to the exponential, note the exponential axis here, to get an idea of the effective fitness of the resistant and the parental cell line. The parental cell lines are the ones that are not resistant to alectinib. And then for that particular well, we can plot on this lower graph the parental growth rate, i.e. the slope of this line, the parental growth rate, i.e. the slope of that line on the x-axis, and the resistant growth rate, the slope of this line on the y-axis, and that gets us some particular point in this space. And then just repeat that again for another experiment, maybe with different conditions. Maybe this one has some other cells in the mix too. and just repeat that many, many times for 4 different conditions: alectinib, and some cancer-associated fibroblast presence, DMSO, and DMSO and cancer-associated fibroblast. So you get this big data set of where each point in the lower graph corresponds to a well in a 96 well plate, and the location of it corresponds to the growth rates of the resistant parental cells. There's something about opacity, which I'll mention in a second. But already from looking at this data more closely, we can already start to see some interesting things, which we wouldn't know if we didn't look at sort of mixed culture, co-culture experiments where we see things together. In particular, we see this condition, DMSO, so there's no drug here, and yet all of the points are above the diagonal of equal fitness for parental and resistant. So, i.e., we're not seeing a cost of resistance. Yet if we looked at just monoculture, that's not what happened. If you look at monoculture and you look at parental versus resistant monoculture, those look like there is, it's neutral. And yet if you plot them in co-culture, then you see actually for any particular mixture of them, it's actually always the case that the resistant beat out the parental. And in general, you start to notice a trend in opacity where more opaque points are sort of up on the diagonals and the less opaque points are down here. And opacity in this case means the initial seeding proportion of parental versus resistant cell, i.e. the fraction of parental cells in the initial mix. And so there's a better way to visualize this.
Before you go on, could you just orientate as to what are all of the points? Are they replicates? Are they like how are you working out the squares, the blue squares, for example?
Yeah. Okay. So let me go to which one I didn't draw on. Let's go here. Let's go back one more. So each point, let's say this one here that I drew an arrow to, that point is a particular well on a plate that had some initial fraction of parental cells, where the initial fraction of parental cells is represented by the opacity of the point. So in this point is kind of like half opaque. So let's say there's about 50% parental cells, 50% resistant cells. And the color of the point represents if we added CAFs, cancer-associated fibroblasts, to the mixture as well. So in this case, blue means that the experimental condition also has cancer-associated fibroblasts in it. And the fact that it's a square versus being a circle means that alectinib is present. So this point means there was a mixture of about 50-50 parental and resistant cells in the presence of alectinib and in the presence of cancer-associated fibroblasts. And then the X position of this point is the measured parental growth rate in that particular well. And the Y position of that point is the measured resistant growth rate of that particular well. And so each one of these points is a particular experiment, basically.
Okay, thanks. I was just worried that was lost on some of the audience.
Mm-hmm.
Artem, this is Mohammed Zahid from Heiko's Lab. Actually, I had a question about the interpretation you're making on that DMSO and it being above the diagonal. And you said, well, there's no drug and there's no cost to resistance then. But couldn't it be that, I mean, it's a vehicle, but DMSO itself has some toxicity. So couldn't that interpretation be that that cost of resistance is, say, resistance to DMSO toxicity or something like that?
Yes. So you could say that we did our DMSO incorrectly, and that our DMSO doesn't actually correspond to no drug. That would be a great conversation to have with a cancer biologist. For me, DMSO is the best that our experimentalists could come up to a no drug setting.
Okay.
Yeah.
My understanding has always been that DMSO, like it has to be inherently looked at as some degree of toxicity just based on its solute property or solvent properties and stuff. But anyway. Yeah,
the parental cell lines have had time to adapt to DMSO for about the same amount of time as the as the resistant cells had time to adapt to alectinib and stuff, like their lineages. So there had to be a very careful explanation as to why the cells that were adapting to alectinib became more adapted to DMSO than the cells that were adapting to DMSO.
Okay.
But maybe that's a conversation we can have offline.
But like at these concentrations, DMSO doesn't really have much of an effect. This is a very low concentration of DMSO, and if we really want to look at the effect of alectinib, we have to use vehicle control rather than, because again, like it's possible that DMSO changes something, but the alectinib is the real drug, and there need to be an adequate control. So vehicle control is sort of the golden standard of experiments.
Right. I was just wondering in terms of interpretation, but sure. Yeah, I don't know what the level is and how much counts it. So, okay, thank you.
Yeah, thanks, Andre. That's the cancer biologist you should ask about this, by the way. Yeah, so sorry, back to this. One of the points that I was talking about is that there's this difference in opacity between the points, and that's really hard to read because hard to read what opacity means from one of these charts. So let's replot our data, same exact data from before. Let's replot it with the opacity made more apparent. Now, for each condition, we're going to plot on the x-axis the initial proportion of parental, i.e. what was represented by opacity in the previous slide. On the y-axis, we're going to plot the growth rate. And when we plot a point in cyan, that's for the parental. And when it's in magenta, it's for the resistant. And so we start to get these fitness functions. This mapping between initial proportion of parental cells to the growth rate in the corresponding mixture. And as you can see, lines aren't a perfect fit, but linear fitness functions do provide some fit to this, to the sort of variance that we're seeing over the initial proportion of parental cells in the mixture. And so we can visualize these fitness functions as a game. In particular, any fitness function is then can be then re-expressed in this form as a gain function, i.e. as the difference between the two fitness functions, and that then becomes a point in game space, where game space is an abstract space spanned on the x-axis by the relative fitness of a resistant invader, and on the y-axis by the relative fitness of parental invader. So each one of these pairs of lines from this figure become a point in this game space corresponding to a particular payoff matrix. And what we can see here is that we have 4 different measured games. They're all quantitatively different from each other. You see some little error bars drawn on those points, and they're not overlapping. But more importantly, they're qualitatively different from each other in the sense of this game space, the x and y axis divide this game space into 4 different dynamic regimes. So if you're in the top left up here, where no games were measured to be in the top left in our case, the parental cells will always invade the resistant cells, no matter how many there are. If you're in the bottom left corner, there's a bifurcation around some central point, where exactly where the central point is will depend where you are in the game space. But around the bifurcation, things go towards either all parental or resistant, depending on if they started above or below the bifurcation point. In the top right corner, where we see the DMSO and CAF measurement, we have an internal fixed point. And if you're above that internal fixed point, you have more parental than that, then you'll go down towards the fixed point. And if you're below that fixed point, i.e. you have more resistant cells than the fixed point, you'll go up towards the fixed point. And finally, in the bottom left corner, you have dynamics where resistance always wins out. And the point of these 4 games is that one of them is not only quantitatively, but also qualitatively different from the other 3. It has a different kind of dynamic, and you can kind of see with the insights what that looks like over time. And why is this interesting? It's interesting because it helps us to put on a firmer footing sort of an intuitive thing, an intuitive axiom that we used in a lot of EGT work. In a lot of EGT work, we kind of think of a therapy as potentially transforming the game. And then afterward, because the game is transformed, the selective pressures are different, the population goes to a different point. And so in this particular case, we can see things like if we take a DMSO plus CAF, which was our surrogate for a patient, this is all done in vitro systems, but it was in some sense our surrogate for a patient before treatment. If you hit them with alectinib, it qualitatively changes the kind of game that's being played. On the other hand, if you have a DMSO patient, or if you have a DMSO plus CAF and you eliminate the CAF somehow, you also qualitatively change the game. So the idea is that treatment can change the game, not just specifically target one type of cell. And so since these two,
Artem, if I can just dive in with a quick question. This is Joel.
Yep.
Should there be included in this density dependence? So sometimes when you have games and there's both density and frequency dependent effects, if you ignore the density effects, it can appear to be game theoretic even when there's no frequency effects, and vice versa.
So these were all seated at the same density.
But were the densities changing with time as you were measuring these? So at the point of measuring these fitness effects, densities are also presumably changing as well as frequencies?
So when you do the analysis of this carefully, even if you did it with different densities, you can imagine the small time window over which we measure them as time epsilon, basically. It's not actually that short, it's 5 days, but the whole point is that small time to get recordynamics out corresponds to the smallest time, right? It's the delta t step, right? So from that, so if you're actually writing down the fitness function afterwards, it would be your initial seeding proportions or densities would be the inputs to the fitness function. So even though they do change in frequency and density over that short time period, we're using that change to get that delta change and mapping it in the fitness function to the initial proportion and frequency. Initial proportion and density. So if you wanted to study density dependence with this sort of setup, you would want to seed your initial cells at different densities. And you definitely could do that, and that'd be interesting, but it would require you to do a lot more experiments. So we already have to do a lot of experiments just to get a number of different proportions at the same densities. You'd have to do even more experiments to do also across with different densities.
Right. No, Artem, only in in the world of ecology, what you're doing is what are referred to as DeWitt experiments. And these are experiments where people varied frequencies but not densities to try to investigate things. So there's a fairly lengthy literature that kind of explores the consequences of examining things in frequency space versus just density space. But anyway, thank you. No, your response was really good. Thank you.
Great. Thanks, Joel. So since the two different differences between these game types is that the ones in the top right corner have have a fixed point, while the ones in the bottom right don't have a fixed point. Well, let's see that in actual cases. So here you see time on the x-axis, proportion of parental cells on the y-axis, and I've color-coded each single line in there is a single individual experiment, basically. And I've color-coded the experiments based on if there's significantly fewer cells, significantly fewer proportion of parental cells at the end of the experiment than at the beginning, then that line is coded in magenta. If there's significantly more parental, i.e. the parental proportion is increasing, those are color-coded in cyan. And if statistically indistinguishable proportion at the start, at the end, then that's color-coded in black. And so again, you see what you expect, which is with DMSO, alectinib, and alectinib plus CAF, which are the games that you expect to see in the bottom right corner. You see it going towards all parental, while in the DMSO plus CAF case, you see it going towards an internal fixed point. You see some upwards movement here and downwards movement there, while here you're only seeing downwards movement, downwards movement, downwards movement. So if you want to see the fixed point this way. And so this whole process is what we like to call the game assay. So you start with, a specifically designed experiment that you then, from which then you get measures of fitness. You then plot those fitness functions. And from those fitness functions, you get points in game space as a summary. So in some sense, we haven't actually done anything. We've just summarized our data as a little matrix instead of, you know, the 60 gigabytes of images that this started with. But I do think this is a useful technique. So I think there's a lot of cases where in your own work, you could, you know, along with whatever other outputs and measurements you're doing of the data, you could already also produce a game point corresponding to your measurements. And those would be very useful to us game theorists because then we'd have more examples of actually measured games as opposed to our intuitions. And so if you wanted to do the game assay at home, so game assay at home, all you need is a system where you initially seed your experiments. So suppose these are little wells that you seed, or maybe you're more fancy and you're growing organoids or something. Each one of these is going to be a different, each box a different experiment. And in each experiment, you seed a different proportion of one cell type versus the other one. So let's say in here, you seed all of proportion of cell type 2. In here, you seed 90% cell type 2. Here, you seed 70% cell type 2. Here, you seed 50% cell type 2. Here, you seed 30%, 10%, and none. So as long as you're doing that kind of experiment, that's the experimental part of the game assay. Just doing these, in this particular case, 1, 2, 3, 4, 5, 6, 7 experiments, but you can imagine having more fine-grained resolution. You can then plot the resulting growth rates that you measured from each experiment as points, where the x-axis is parental proportion of type A proportion, ranging from your first experiment, which was 0, to your final experiment, which was 1. And you can imagine plotting the measured growth rates of the two types. And after you've plotted them all in this graph, you then look at the line of best fit. Those lines of best fit then have an equation, which you could write down in the form of, you know, if this is P as the proportion, you could write them in the form of AP plus 1 minus PB for this top line. Say that's the type A line, the type A fitness. And this is the type B fitness. And for the type B fitness, you could write down as CP plus 1 minus PD, where you get these coefficients from your line of best fit. And then if you plot this resulting matrix, A, B, C, D, you have the corresponding effective evolutionary gain. And so there's plenty of experiments that already do these kind of measurements. And they don't usually report this as an output. But if you did report this as an output, even the supplementary materials or something, that would be really great information for game theorists, because then instead of always sort of guessing what we think these interactions are happening, what these interactions look like, we could have sort of a collection, a catalog of interactions that we already know about that have been measured. And then we can compare what we're getting from our simulations to what has actually been measured with exact, you know, final measurements. And so this lets us do a sort of empirical abstraction over games. The idea is that any particular effective game can be implemented by many, many different kinds of reductive games combined with different kinds of, let's say, spatial structure. So this is a particular effective game. I think this is one of the games that we measured in the previous experiment. And one way you could get that game is you could take the same exact game and play it in a perfectly well-mixed population where everybody interacts with everybody else, assortment is random, etc. And then it would just be exactly the same game, the reductive game and the effective game. In the same sense that when you're thinking about simple physical models, you can think of temperature, which is an effective property, as being just average kinetic energy, which is a reductive property, let's say. On the other hand, maybe you have some spatial structure. Maybe your cells actually live in a 3 regular random graph. Well, in that case, a slightly different reductive game, notice different coefficient here and here, would map to the new effective game. In fact, it's not just slightly different, it's a different type of game. So up here, this is a leader game, while down here, this is a hawk-dove game. So it's a different kind of reductive game that you would have put in for your intuitions, but when you added a little bit of spatial structure to it, you get this same effective game. And similarly, in our experiment, there is some spatial structure that we just don't really study very carefully. So we don't know what the reductive game is. We don't know what the transformation is exactly. But after the unknown reductive game is mapped through the unknown transformation, we do know what the resulting effective game is. So that's the sense in which effective games are an abstraction that eats up both the concept of space and the reductive interaction that happens.
Artem, this is Joel just jumping in for a moment.
Yep.
I see, and it's super cool, the fact that the kind of effective game appears quite differently when you have the spatial structure. Could you also account for this by simply defining your game as occurring with non-random interactions? So generally, usually in a matrix game, you assume a well-mixed population, random interactions. What if you simply frame the game as having some probability in which you're not actually, you've got some sort of association, disassociation, so individuals are not interacting randomly? Could you also then present it in that fashion as well?
Yeah, I would call that a particular kind of spatial structure. And in fact, I think you can generalize the Otsuki-Novak transform to apply, because if you actually look under the hood, what does it actually happen in the transform? It just talks about a bias in self-interaction terms.
Right.
And there's a function that maps that bias based on the actual proportions that are happening and the degree of the network. So yeah, you can definitely think of it in that terms, or any of your favorite population, or any of your favorite population structure models. Yeah, so definitely that's the case. But the point is that there's many different choices of, that you could have made for how you think about population structure.
Sure. Yeah.
That when combined with different games give you the same output at the effective level.
Right. Got it. Thanks.
Yeah. Yeah. And so that's all I want to talk about. And yeah, thank you guys for listening.
Nice. Thanks, Artem.
Yeah, thank you.
Yeah. I saw that there was maybe questions in chat, but I didn't look at them as I was talking.
Yeah, obviously people might want to raise those points as well, but I know that some of them are very sort of specific of the whatever slide you were talking about. That might be just about clarifying whether certain points translucency has a meaning or not. So if people need to ask these questions again, I think this is the time. Don't be shy. Or any kind of question, really. At this stage, this is the time when we ask questions to Artem. I know that people were not exactly holding until the last minute, but...
Hi, Artem. I had a question on slide 34. You had the show me the game slide, and you had basically a bifurcation in the bottom left quadrant between and resistant. So does that bifurcation follow the like the identity diagonal?
Identity diagonal
meaning so like, basically above, yeah, exactly. So above the identity, you have a higher relative fitness of parental, and lower you'd have a higher relative fitness of the resistant invader.
So let me see if I can find an empty slide with this. No, they all have drawings on them. Okay. No, so the exactly which line you lie on will determine where the fixed point is. So at the identity line that you're talking about, that'll mean that the fixed point around which the bifurcation happens will be at 50-50.
Oh, okay.
While if it's a different slope, you can imagine, in fact, it really is like just the slope of that line that determines the where the bifurcation point is. And then how far out you are from the origin along this line, right? So if you're here on the line versus here, I'm not sure if you can still see what I'm drawing.
Yeah, you're good.
Will determine how sharp the selective pressures will be. So once you fall off that fixed point a little bit, if you're up here, then you'll only slowly move away from the fixed point. While if you're all the way out here on the same line, you'll move very quickly away from the fixed point.
Right.
So
it'll determine your speed of convergence to the dominant fixed point, whichever one you're going to. Okay, thanks.
So I have a couple of points. I just wanted to, I guess many people probably know this, but it's worth stating. This is one of the first, if not the first, game that was calibrated or parameterized with data. Is that correct?
Yes, directly. There are some more indirect measurements that have been done previously.
So Artem did have something on PNS some years ago that I know that he got some critiques for the way he approached it, but he was using experimental data too.
Yeah. So in Arkadi's case, there's this nice paper, which was actually an inspiration for us. Mm-hmm. He basically did all the steps that we do here to measure the game. It's just that he then did not report the game. He then went back to this qualitative segment of saying, oh, look, I have a more than, because his point was that this is going to be a nonlinear game. And he was trying to show, look, I have more than one fixed point in the resulting dynamics. And in fact, if you go back to some of the virus and microbe literature, they actually ended up, and some of this I wasn't actually aware of at the time that we wrote this. They actually, because the actual, where's the slide where I described what the game assay involves? The game assay involves actually very little in terms of experiment, right? It just involves doing this experiment, a series of series of different co-cultures with different initial proportions. And obviously we're not the first ones to do different initial proportions of co-cultures.
Of course,
that's been done before. And it's been done before in game settings. It's just people have never gone the direct step of saying, if we make the experiment this way and we plot the data this way, it can be then summarized as this game. That's the new step in our work, I guess.
Sure, and I think that's one of the reasons why I bring it up is that there's obviously utility emerges from this ability to map it onto this game and then reflect on how these different sort of experimental pieces give you different mapping or different values of that mapping. And so you can compare them directly, right? But obviously you're fitting, and I think that one of the reasons why I brought up all those replicates is there is a lot of variation and you're looking for very general trends. And that's what we have to do when we're doing experiments, right? But is there anything that meaningful in that variation?
Yes, so I completely agree that variation is very important and the noise is very important. Yes. And so if I didn't go into it in the talk because I think people would definitely fall asleep, but I do go into it much more in the paper. We're actually very careful about propagating our errors. And I think that's very important. Like one of the things that bugs me sometimes is I don't see error bars on things that people measure. We're actually very careful about from the error we have on our initial measurements of growth rate to, propagating that to the error on, these lines of best fit. They have an error on them as well. I'm not visualizing it here, which then become the error bars on the actual game points. So it's very important for that variance to be propagated all the way through. And you can imagine that if the variance was very high, so we can look at this DMSO calf point here that maybe I'll erase some things around it so that we can actually have some, whoops. I seem to not be able to erase. Okay, erase some stuff. Around this point, you can imagine that if we had more variance in our experiment, and if, let's say, linear fits weren't a good fit, then these error bars would expand. And once they expand to the point that they're not confined to a single quadrant, well, then in this case, you can't make any conclusion about the type of game because it could be like any dynamic. Your error bars are just too wide to have any confidence in your result. So in this case, we did get lucky in that the experimental system we happen to have, happened to give us nice error bars at the end. And it might not be the case that you always get that. And in that case, you might want to look into other cases. So there was a study done in some bacteria that colonize the guts of Hydra. And there, for example, the linear games that we fit here would not be, with their data, good fits. You would really want to go to quadratic games. But then quadratic games, you would need a different game space to plot them in. So yeah, I think we should definitely handle variance carefully, make sure we propagate all our errors through, and make sure that if we are making a conclusion about two dynamics being different, that the resulting error bars in the proper space of dynamics don't overlap.
Yes. And so I guess a point related to that is this is implicitly pairwise interactions you're looking at. And obviously what's beautiful about those experiments is they're probably also nicely pairwise interactions. But if we then try and think about this in the context of, say, an actual patient who's been treated with this drug, pairwise interactions might not be right in that setting, right? Or they might not be the only component we need to care about.
Yeah, no, I completely agree. I think I personally right now see this mostly as a thing to perfect and work about in these very well-controlled in vitro systems where we can actually make the measurements very carefully. I'm not completely sure how we would make an in vivo measurement, or at least in a patient. I can imagine how we could do it in a mouse, but it's hard to imagine how we would do these kind of measurements of games in a patient, at least right now.
But obviously, as people who've done measurements of sort of, say, fibroblasts interacting with tumor cells using some sophisticated sort of imaging cranial assays, like the guy that's, his name escapes me at the Crick. He's done some cool stuff with melanoma, where he's looked at